Thank you to the International Eczema Council for the honor of being part of such an outstanding team in putting together the review on human models of atopic dermatitis, and for the opportunity to share the contribution of human equivalent models, or those models in cell and tissue culture. There are several types of human equivalent models, including 2D culture, 3D human skin equivalents, 3D human epidermal equivalents, and ex vivo explant cultures. These models can be built in many different ways to study atopic dermatitis pathogenesis and therapeutic applications. Moreover, these models can be used in tandem as complementary tools to study this disease. Some key characteristics of atopic dermatitis recapitulated in these models include decreased barrier function, hypogranulosis, increased proliferation, spongiosis, thickened stratum corneum, alteration of key differentiation proteins, increased immune activity of keratinocytes, changes in immune cell profiles, and the interplay of different skin cell types in co-culture. Here I will present some advantages and limitations of each model, as well as some primary data from my laboratory. Let's explore 2D cultures first. Perhaps the biggest advantage of 2D models is the potential for design of high throughput screening assays for drug development or validation of novel therapeutic targets. In addition to keratinocytes, other skin cells can be cultured in 2D and assessed for atopic dermatitis-like characteristics, including immune cells. Co-cultures of different cell types are useful to define heterotypic cell interactions. Keratinocytes and other skin cells remain responsive to Th2 cytokines in 2D culture. A minor limitation of this and other human equivalent models is possible limited access to fresh tissue or primary cells. However, an increasing number of companies provide these cells, as well as many good collaborators. An inherent limitation of working in 2D culture is the limited barrier function. Barrier function can be measured by transepithelial electrical resistance, or TER. In this assay, cells are grown on the membrane of the upper chamber of a transwell plate system. Electrodes pass an electric current through, and the instrument reads the resistance of the cell layer. Higher resistance indicates greater barrier function. While the barrier function can be measured in 2D, 3D systems have significantly higher resistance. This is largely due to the fact that submerged cells do not stratify as a tissue and do not express or organize barrier proteins as in 3D cultures. But we must remember that primary skin cells are propagated in 2D for downstream 3D applications, such as 3D human epidermal equivalents and skin equivalents, which I will talk more about now. Here is a view from above of a 3D human skin equivalent cultured at the air-liquid interface. These cultures are fully stratified with a dermal compartment, are responsive to Th2 cytokines, and are amenable to various cell combinations for co-culture. Application of 3D human skin equivalents can be limited by individual laboratory expertise or the time investment required to establish these cultures. But again, if a good collaborator cannot help fill this gap, there are commercial options. To create these cultures, a collagen hydrogel is embedded with fibroblasts, and keratinocytes are plated in a confluent monolayer on top. After a couple of days in submerged conditions, the cultures are placed at the air-liquid interface. This environmental change alone induces keratinocyte stratification and differentiation. Of note, the morphogenesis of the epidermal compartment can be monitored over time. The morphology of 3D human skin equivalents changes after exposure to interleukin 4 and 13 in combination. And this change varies depending on the maturity of the cultures. In 3D human skin equivalents cultured for 6 days at the air-liquid interface, there is spongiosis in the spinous layers, hypogranulosis, and loss of cornification. After 12 days at the air-liquid interface, exposure to Th2 cytokines induces a disorganized basal layer and loss of attachment to the basement membrane, hypogranulosis, and at this time point, there is a thickened cornified layer. Perhaps the most powerful 3D human skin equivalent tools will come from optimized protocols for incorporation of immune cells in co-culture models. We are optimizing 2 types of co-cultures. The first incorporates T cells into the media of the 3D human skin equivalents. In the absence of T cells, 3D human skin equivalents stratify normally, and there's characteristic expression of loricrin in the uppermost epidermal layers. The same phenotypes are noted when T cells are added to the cultures. However, when T cells are polarized, in this case as Th1, there are drastic changes in epidermal morphology described by a thickened stratum corneum, hypogranulosis, and a loss of loricrin expression, suggestive of a decreased barrier function. The other model we are trying to optimize in our hands is the incorporation of immune cells in the dermal compartment or the collagen hydrogel of 3D human skin equivalent cultures. So far, we can show the survival of CD3 positive T cells in the dermis, lining up just below the basement membrane and epidermal compartment. We now turn to a less complex model, the 3D human epidermal equivalent, comprised of primary keratinocytes grown in a transwell system. These are also fully stratified, but take less time and expertise than 3D human skin equivalents. You can assess barrier function using a couple of different methods in this model, and co-cultures are possible. But we must remember that there is no dermal compartment in the 3D human epidermal equivalent. A key application of 3D human epidermal equivalents is the ability to assess barrier function. As primary cells in this culture system are responsive to Th2 cytokines, we see a significant decrease in resistance after treatment with interleukins 4 and 13. Another experimental advantage of this model is the ability to stain whole tissue, in this case, whole intact epidermis. Here we see an en-face view of 3D human epidermal equivalent stained for occludin to demarcate tight junctions. In these control cultures, we see completely or nearly completely enclosed occludin rings. In contrast, 3D human epidermal equivalents exposed to Th2 cytokines show punctate staining and loss of occludin rings. Finally, we will discuss ex vivo explant cultures. These are whole tissue with many cell types, keratinocytes, fibroblasts, and immune cells, but also remember, there are blood vessels and nerves. These whole tissues are responsive to Th2 cytokines, and explants derived from biopsies of patients with atopic dermatitis retain their phenotype. However, these cultures have a limited lifespan compared to other 3D models. This is a representative image of control skin tissue cultured at the air-liquid interface for 7 days, compared to tissue exposed to interleukin 4 and 13, where we see spongiosis in the stratum spinosum and movement of cells in the dermis toward the epidermis. Home outstaining can also be done on ex vivo explant cultures, but in this case, I'm showing a cross-section of occludin staining. In contrast to control cultures, there is a mislocalization and overall loss in occludin, indicating loss of tight junction barrier integrity in cultures treated with Th2 cytokines. Now, it's not just the living part of the culture that can be of interest when studying the biology of atopic dermatitis, but the culture media itself can hold answers to questions, especially those asked about secreted factors of skin cells. Here is an example of cytokine arrays exposed to control media or media from explants treated with interleukin 4 and 13. As expected, we see increases in IL-4 and 13 as it was added in the media, but also increased expression of proteins involved in vasculature, such as VCAM1 and VEGF, which have been shown to be overexpressed in AD or correlate with disease severity, respectively. We also find an increase in CCL17, which has been shown to induce migration of Th2 cells into AD lesions. So in summary, we have several in vitro models to study atopic dermatitis and other inflammatory skin diseases. There can be different inputs for these models, including normal keratinocytes. These keratinocytes can have gene modulations. We can co-culture different cells in these models. We can use patient-derived tissue and cells for the establishment of these cultures or the co-culture. And we can use different cytokines to induce atopic dermatitis-like phenotypes. The output or the data derived from these includes assessment of epidermal morphology, the extent of differentiation, focusing on barrier, cytoskeletal, and adhesion proteins, keratinocyte and immune cell factors that are secreted. We can assess barrier function through TEER, looking at lipid profile, as well as paracellular permeability. And we can also hone in on key transcript and protein changes to directly assess changes in signaling events in the immune and barrier systems. And once again, I would like to thank the International Eczema Council.